Plant Physiology Breakdown, Photosynthesis and Respiration for CSIR Net Life Science. Hello viewers of Biotechnica. I believe that you understand how important these two topics are in your preparation for the exam. Let me try to make it a little bit easy for you. Also to note, the entrance exams having the same topic in their syllabus is CSIR Net Life Science, GATE, BT, Excel both, Biotechnology and Life Sciences, ICMR, TIFR, ICAR and DBT. So if in case you are preparing for any of these exams, these are very fundamental concepts and will be applicable and present in the syllabus of all this life science competitive PhD entrance exams. Now, if I have to simplify the two processes, that is cellular respiration and photosynthesis, one occurs in the mitochondria, that is cellular respiration, another will take place in the chloroplast. The mitochondria will be using the food molecules and oxygen, and they would be reactants of respiration, which will be converted into ATP energy, and the products of respiration that will be given out will be carbon dioxide and water. Whereas in photosynthesis, these carbon dioxide and water only will be utilized, and with the help of light energy through some processes, obviously they will be converted into glucose and oxygen. So these are the products of photosynthesis. There are a lot more details in here, but remembering the basic elements of these two processes is very crucial. First, we will look at cellular respiration. This is the overview of the process. As I said, the raw material for cellular respiration is oxygen and glucose. Now, whenever we consume glucose, it is undergoing the process of glycolysis, in which it will be broken down into pyruvate molecules. Pyruvate further is converted into acetyl-CoA, which enters the citric acid cycle, and from the citric acid cycle some reducing equivalents are generated, which is NADH, FADH2, and even there is some substrate level phosphorylation, which makes ATP. Substrate level means the substrate bond breaking will release out some free energy, which can convert ADP and PI into ATP molecule. Okay. Apart from that, even pyruvate to acetyl-CoA, the conversion is done by PDH, pyruvate dehydrogenase complex. This is also releasing out NADH, reducing equivalent. Even glycolysis also releases NADH. So with all this NADH, the reducing equivalents which have the protons and electrons in it. As you all know, NADH has 1 proton and 2 electrons in it, whereas FADH2 has 2 protons and 2 electrons with it when they are in the reduced form. So these are all the reduced form of it. Now these electrons will be given to the electron transport chain. So from the cytosol, the NADH will be coming inside the mitochondria, and anyways Krebs cycle is taking place inside the mitochondria only. Mitochondria, as we all know, it is a double membrane structure. The inner membrane of the mitochondria is thrown into folds, which is called as cristae. The reason is because the electron transport chain is facilitated by some complexes, protein complexes, which have some carriers of electron. So this NADH, FADH2 will be passing on the electrons from carrier to carrier in this electron transport chain, and ultimately they will be generating ATP through the ATP synthase. I will be telling you how. Overall this will be generating 32 to 34 ATP. So the electrons, after passing through electron transport chain, ultimately it will be given to oxygen. Oxygen with hydrogen and the electrons, it is going to make water molecule. Okay. So that is why I said oxygen and glucose will be required as the raw materials for respiration. And since this ATP is produced by the utilization of oxygen, that is why this phosphorylation, as I said, this was substrate level phosphorylation. But this phosphorylation which you generate through electron transport chain and ATP synthesis, it's called as oxidative phosphorylation. Okay. Now going into further more details over here, mitochondria, it is the site of the cellular respiration, as I said. The inner fold of mitochondria is thrown into this cristae. The reason is because we are calling mitochondria as the powerhouse of cell, right? So that's why there should be a lot of this oxidative phosphorylation process taking place. That means we should have many of these complexes which should be present, electron transport chain systems which should be present. So just to increase the surface area, the inner membrane is thrown into fold so that it can accommodate a lot of electron transport chain complexes. The Krebs cycle enzymes are present in the matrix, and this complexes, how they will be, the electrons will be passed from one complex to another complex. In that process, these complexes will be acting as proton pumps. So they will send the protons from the matrix side into the intermembrane space. So outside will be the outer membrane. This is the outer membrane. This is the inner membrane. In between what you see this gap, it is the intermembrane space. So from the matrix through the electron transport chain, NADH and FADH2, they will be present in the matrix. Once they give the electrons to the carrier complexes, they will pass the electrons, and on the other hand they will pump protons towards the intermembrane space. Okay. Now these protons, they have to come back to the matrix, otherwise it will keep on getting accumulated over here. It creates a gradient. This cannot stay as it is. So in order to come back to the matrix, they have to cross the membrane. But the problem is that the inner mitochondrial membrane, it is impermeable to H+ ions. Okay, so these are important facts that you must be knowing. Impermeable to most of the things, inner mitochondrial membrane, but most important here is the H+. So H+ cannot come through the membrane. That is why H+ comes through something which is called as a F0F1 particle, which is the ATP synthase. Now when it comes through that, this protein has this enzyme complex has a proton channel. So when the H+ ions come to the other side, they will be creating or generating the ATP molecules. How? I will be showing you shortly. Next is the electron transport chain. This is what is the electron transport chain complexes I was talking about, which are present in the inner mitochondrial membrane. I already told you that the Krebs cycle enzymes are present in matrix except one of them. Okay, so the one enzyme complex, it is actually present in the form of a complex. It is not present in the matrix, but it is present in the inner mitochondrial membrane. It is embedded in the inner mitochondrial membrane, and that enzyme complex is nothing but succinate dehydrogenase. So this electron transport chain has 4 complexes in it, out of which the second complex is actually an enzyme of the TCA cycle or the Krebs cycle. You would be knowing that succinate dehydrogenate converts succinate into fumarate. This is a step of Krebs cycle, and this is done in this enzyme complex only. As you can see, succinate to fumarate is done, and this particular step in Krebs cycle releases electrons. So we have the production of FADH2 I was telling in Krebs cycle. It is from this particular step only. Now when FAD is released, FAD will be accepting electrons and it will be making FADH2. This FADH2 doesn't have to go anywhere. In fact, it will directly give the electrons to this iron sulfur center. Okay, directly it is going to give the electrons here, and then the electrons are going to get passed on further. What about the NADH? Where will they be giving the electrons? NADH gives the electrons to complex 1. Remember that. This is the first complex, which is called as NADH dehydrogenase. NADH from several sources that we saw, from glycolysis, pyruvate to acetyl-CoA, and even from the Krebs cycle, they will give the electrons to this first complex, in which you have these carriers called as FMN and iron sulfur centers. They will further pass on to ubiquinone. Now you will see that there are separately 2 of these molecules. One is ubiquinone, another one is cytochrome C. So these are called as mobile carriers. Okay, these are not embedded in the fixed position in membrane, but they can actually move from one complex to other. So they are acting as mobile carriers. They will take the electron from one complex and pass it to the other complex. But this flow of electrons within the complexes is a little bit different. First of all, you should know for NADH. Now, what is the flow of electrons for NADH? From complex 1, it is given to ubiquinone. From ubiquinone, it is given to complex 3. Okay, complex 3 gives it to cytochrome C, and from cytochrome C, it is given to complex 4. So you saw that how I have skipped complex 2 when we are talking about electrons of NADH. So NADH does not give the electrons, does not involve the passing of electrons through complex 2. It does not involve complex 2 at all. Same logic applies when we are talking of electrons given by FAD. So FADH2 is the only one molecule produced by this enzyme. Apart from that, any other process in these oxidation steps does not produce FADH2. So the only FADH2 over here, in fact, that is why here the electron acceptor is FAD. Okay, otherwise it would have been NADH only. Just because it is present in this enzyme complex, that is why here the electrons are accepted by FAD. So FADH2 directly gives the electrons to ubiquinone, the mobile carrier, and ubiquinone gives it to complex 3 to cytochrome C, and cytochrome C to complex 4. Now you have noticed that I have completely skipped complex 1 when I am talking about FAD, isn't it? So for complex 1, it does not involve complex 2. For NADH molecules, it does not involve complex 2. And for FADH molecules, it does not involve complex 1, right? And finally, the electrons, they are given to oxygen. Oxygen is the terminal electron acceptor. Okay, oxygen is the terminal electron acceptor, and after combining with protons, it would be making water, right? So this is only possible in case of aerobic conditions in the cell. Only when oxygen is present, this oxidative phosphorylation can take place. Now another thing to notice over here, as I said, when the electrons are passed from complex to complex, from carrier to carrier, I said these complexes are going to pump protons to the other side of the membrane. So as you can see, the other side of the membrane, the complexes are passing the protons, all the complex except complex 2. So complex 2 doesn't pass any protons to the other side because this is an enzyme complex. It doesn't have a proton channel. Okay, but the complex 1 passes 4 protons to the other side. Complex 3 also passes 4 protons, but complex 4 passes only 2 protons. Anyways, here also 2 protons are getting utilized, joined with oxygen in order to form water. So here total of, if you talk NADH, then it involves complex 1, 3 and 4. So total of 10 protons are passed across the membrane when one NADH molecule passes 2 electrons through the electron transport chain. So this is how facts and figures are important here. When one NADH molecule gives 2 electrons to the electron transport chain, a total of 10 protons are transported to the opposite side of the membrane. Now what will happen if there is a excess of protons on the other side of the membrane? This will lead to the formation of a gradient which is called as the proton motive force. Why proton motive force? Because it is having a high concentration of protons on the other side, whereas on the other side you will have a low concentration of protons. Okay, so that is why this side is negative. Since protons are positive charge, this side, which is the matrix side, it is the negative end of the system. So this side is also popularly called as the N side. Okay, whereas towards the outside you have more of positive charge, that is in the intermembrane space. So this side is more commonly known as the P side. All right. So what is this electron transport chain? During electron transport, energy released is used to transport H plus across the inner mitochondrial membrane to create an electrochemical gradient. So this is where electrochemical gradient is formed. Why electro? Because you have the charges involved. And why chemical? Because it involves this H plus. We know H plus is related with the acidity and alkalinity of the solution. That is why we are calling it chemical gradient also. Okay. As I was mentioning, the electrons collected in NADH and FADH2, they are released and transported to oxygen via the respiratory chain. Already what I mentioned is that this happens in the cristae of the mitochondria. Some point to remember is that this was revealed by Eugene Kennedy and Albert Menninger. Sometimes the scientist name questions are also coming these days. So ensure that you are able to relate the scientist name with the kind of discovery or invention that they have done. Okay. Now, if you see one logic in electron transport chain, why the protons are pumped towards the other side, where already there is a higher concentration of protons, but still the protons are pumped from a region of low concentration to a region of high concentration. So obviously this process is not at all favorable. This is not a spontaneous process, passing protons from low to high, but still these complexes are doing so. So question is, which process is driving this flow of protons from a low concentration gradient to high concentration gradient? The answer lies in the power to accept electrons, which is called as redox potential. Okay, so more the redox potential, more easy is accepting electrons. So you must remember that the redox potential of the last electron acceptor, oxygen, is highest, and the redox potential of the initial electron acceptor, that is FMN, it is the lowest. Okay, so it is very difficult to give electrons to FMN, but as it goes down the carrier chain, it becomes more and more easy to accept electrons. So what you can say is that the electron flow is downhill. This is a very important concept. The electron flow is downhill in case of the electron transport chain. Now whenever you have a downhill process, that means it's a favorable process, going from something which is a low reduction potential to high redox potential. It is more easier, right? So if any process is easier, then you would release some amount of energy, and that energy is coupled to the flow of protons. Because these electrons are transported downhill, that is why the protons are able to be transported uphill. So there is a difference of 1.14 volt potential difference between the NADH, which is having, as you can see, redox potential in negative minus 0.32 volt, whereas oxygen, the terminal electron acceptor, it is having 0.816 volt, which is very high redox potential. This entire thing drives the electron flow through the chain. Fine. Already, as I mentioned, the respiratory chain consists of 4 large multi-protein complexes, 3 being proton pumps and 2 are mobile carriers. And there are also some prosthetic groups in these protein complexes. These are nothing but the carriers which we saw, FMN, FAD, the electron carriers, iron sulfur centers, 2Fe, 2s, 4Fe, 4s, heme A, iron protoporphyrin, heme C, etc. So the flow of the electrons, by the way, these are the corresponding complexes and their prosthetic groups. This is a easy target of match the following question. Okay, so easily they can ask you which prosthetic group is present in which of the complexes, and in fact which inhibitors can inhibit these complexes. Even that is a popular question of CSIR. Maybe that we can discuss in some other video, or you can attend the classes of Biotechnica where we discuss all these concepts in further much more details as is required for the exam. Now, I was talking about the redox potential. You can see we start with NADH giving off the electrons from the complex 1. So, the redox potential over is in negative. As I said, minus 0.32 volt is there. This is given in millivolt, by the way. So, from here, you can remember the flow of electrons. That is, from NADH, it is given to FMN. From FMN, it is given to FES, then to coenzyme Q, then to cytochrome B. So this is complex 3, which is also called as cytochrome BC1 complex. It is also called as cytochrome C reductase. So both the names you should be knowing. And from cytochrome B to FES, cytochrome C1, you can see that constantly the redox potential is getting increased. Okay. Finally, cytochrome C1 to cytochrome C, then to the complex 4, which is also called as cytochrome C oxidase. And there we have this copper, cytochrome A, copper B, and finally the electrons are given to oxygen. Okay, so this leads to the formation of water. Also notice the free energy change. You will see that the free energy is high before, but it slowly comes down as it reaches the terminal electron acceptor, which means that this flow of electrons is a spontaneous process because the free energy is decreasing, free energy is negative, so it is an exergonic process. That is why it is able to transport the protons to the other side of the membrane. Okay. So oxidative phosphorylation is the process by which energy stored in NADH and FADH2, it is used to produce ATP. The first step is the oxidation step, where we have the electron transport chain. So here the NADH and the FADH2, they will give the electrons to oxygen, thus regenerating the NAD plus and FAD for the rest of the cycles and pathways. And the second step is phosphorylation, where the energy released from here by the passing of electrons is used to drive the formation of ADP and PI to ATP. Now this is done on the ATP synthase molecule. So this was the electron transport chain till here. Now after this we got a gradient of proton here in the intermembrane space, from where the protons will try to come back to the other side of the membrane. Okay, but as I said, they cannot cross the membrane. Inner membrane is impermeable. They have to do so by crossing through the ATP synthase molecule. Okay, now ATP synthase has two parts in it. One is the F0 particle embedded inside the membrane. Another one is the F1 particle, which is protruding towards the matrix side. And F1 particle has the binding sites for ADP and the PI, which are raw materials for forming ATP. And as H plus ions pass through this ATP synthase proton channel, this F1 particle and F0 particle, it is having a rotor in their structure. This rotor rotates, which induces a conformational change by which ATP will be synthesized. Okay, so this is how ATP is synthesized. We have the membrane electric potential at this side, high hydrogen ion concentration. So that is how we call this process as chemiosmosis also. And we have an electrochemical gradient over here. Okay. During oxidative phosphorylation, the energy generated during oxidation, proton electrochemical gradient, is coupled to ATP production, which is ADP phosphorylation. So since H plus transport results in the electrochemical gradient, this requires an intact inner mitochondrial membrane so that the gradient is formed properly. If it is somehow this gradient is dissipated, so we have some inhibitors of electron transport chain which are called as uncouplers. Because of that the gradient is dissipated, then this electron transport chain cannot take place properly. Okay, because the electron transport chain cannot take place properly because this process is solely based on the proton motive force. Now there will be some antiporters and transporter, ATP, ADP antiporter. The ATP formed in the matrix will be going out through this and the raw material ADP will be coming inside the matrix. As I said, inner mitochondrial membrane is impermeable, so most of the things need to be sent through the transporter. And even phosphate transporter PI will be sent inside and OH ions in turn will be sent out, and in fact H plus ions will also be sent out for the same thing. Okay, translocation of H plus ions during electron transport was happening. Now this proton needs to come back. So this proton motive force energy released by flow of H plus down its gradient is used for ATP synthesis. Inner mitochondrial membrane will be impermeable to H plus. So it goes through this ATP synthase, right? So H plus channel that couples energy from H plus flow with ATP synthesis. With every 3 protons transferred from the ATP synthase, 1 ATP molecule will be generated. Okay, so by this logic, since from NADH we had 10 protons, so roughly 3 ATP will be generated from 1 NADH molecule, and from 1 FADH we had roughly 6 protons, so 2 ATP will be generated from FAD molecule. There is some updated detail that some sources will say that 4 protons are transported from the ATP synthase, then 1 ATP is formed. In that case, you will produce 2.5 ATP per NADH molecule and 1.5 ATP per FADH2 molecule. but there is still a discrepancy in this information, so answer the questions accordingly. Flow of H plus down its concentration gradient releases energy to fuel ATP synthesis, which requires ADP and inorganic phosphate, which are transported into the matrix. Okay. Coming to photosynthesis, which is synthesis using light. So basically here we have the carbon dioxide from the atmosphere, water from the soil, which will be converted into glucose, food for the plant, and also oxygen, which will be released outside for us. Now this photosynthesis and cellular respiration together, this is actually a classic example of a redox reaction. So photosynthesis harnessing light energy, plants use sunlight, carbon dioxide and water to produce carbohydrate with oxygen as a byproduct. This overall chemical reaction summarizes the entire process. Usually glucose is considered as the carbohydrate made, so that's why this will be the entire balanced reaction. 6 molecules of carbon dioxide, 12 molecules of water with light energy will produce 1 molecule of glucose, 6 molecules of water and 6 molecules of oxygen. But a better summary of this is that you have two different set of reactions. One is the light reactions, which involve the use of light, not that it takes place in the day, but it uses the light. Yes, light is in the day. And another one is the dark reaction. Dark reaction doesn't take place at night. It's just that it doesn't utilize light energy over here. So with light energy, water is converted into oxygen, and the electrons that is released from here, it goes through a series of electron carriers, just like how we saw in oxidative phosphorylation, and it will be harnessing it into chemical energy. So equivalents such as ATP and NADPH will be generated, which then enter the Kelvin cycle, and they will be using this chemical energy to form the glucose molecule. Now while the light reactions take place in the thylakoid region of the chloroplast, whereas the dark reaction take place in the stroma of the chloroplast. Okay. So photosynthesis in plant, it takes place in the chloroplast. So yes, this is the stroma. Even chlorophyll is a double membrane structure. Inside the lumen of the chloroplast, which is called as the stroma, we have some stacked layers like this. So these dark reactions, they occur in the stroma, as I said, and these stacked things, these are called as thylakoid. Okay, when several thylakoids will be stacking on top of each other, then they form a grana, or which we can call granule lamellae. And stroma also has some connections. These are the stromal lamellae. The light reactions occur in the thylakoids, that is the PS2, and stromal lamella will be having this PS1. Okay, these are the electron passing processes I will be showing you. And yes, thylakoid membranes appear stacked like coins, but in fact are highly folded, and they have a well defined interior also. So inside of this thylakoid is called as thylakoid lumen, and exterior with respect to the stroma. So outside is actually facing the stromal membrane. So the basic concept of energy transfer during photosynthesis, yes, we have different pigment molecules involved in photosynthesis. So when the light energy, it is first captured by accessory pigment molecule. The main pigment molecule is chlorophyll, obviously. So several accessory molecules, they comprise something called as an antenna complex, which will be passing on the light energy in the form of photons to the reaction center. And from the reaction center, water will be undergoing photolysis, that means breakdown by light, and they will release the electrons which will enter the photosystems further. Okay, so the two processes in photosynthesis that capture light and produce energy rich compounds that are used in carbon fixation, they are termed as photosystem 1 and photosystem 2. If you notice over here, this is first we have the photosystem 2. This is the reaction center, which is termed as P680. Surrounding it are certain accessory pigment molecules. So finally the reaction center will get the light P680. After getting activated, it will become reduced over here. Okay, that means it will get the electrons over here, but electrons, they will not keep it with itself. It will keep on passing the electrons through the electron transport chain. There will be a series of carriers, after which it will be passing on to the photosystem 1. So here the reaction center, this 680 and 700 are the nanometer, the wavelength of light that they will be absorbing. So after getting the electron, this goes into an activated state, that means in a reduced state. But again here also they can't keep the electrons to itself. They will keep on passing the electrons, and finally the electrons are given to NADP. Thus you get the generation of NADPH. And as I also said, from this processes ATP is generated. How is ATP generated? Now when the electrons are passed, simultaneously protons are also transported in the same logic and manner that we saw in oxidative phosphorylation. So electrons flow downhill, and that's why protons are pumped uphill. Here also gradient will be generated, and there is a separate chloroplast F0F1 particle. In chloroplast also it is there, ATP synthase. From there ATP will be generated. So these two processes, photosystem 1 and photosystem 2, they are linked together in which is called as Z scheme of photosynthesis. Okay, so because this process looks like a Z scheme, there is cyclic and non-cyclic photophosphorylation, which maybe we can discuss in some other video. The Z refers to changes in redox potential of electrons, and note that the PS2 comes before PS1 in this particular scheme. Okay. So we have this strong reductants and weak oxidant. Know that part also. The wavelength of maximum absorption is in the red region for photosystem 2, P680 nanometer, and P700, the wavelength of maximum absorption is in the far red for P700. Okay. Next is ATP synthesis by photophosphorylation. Now here the concept is same, just that the location is different, because these complexes PS1 and PS2, they are present in the thylakoid membrane. So this is the thylakoid membrane. From here the complexes, they will be passing the protons inside the thylakoid lumen. Okay, so that's why thylakoid lumen has positive charge. Thus it is called as the P side of the apparatus. And this is the chloroplastic CFO and CF1, from where if the protons are passed, then one ATP molecule is produced over here. Okay. Now notice that the F0 particle is protruded towards the stroma. That means stroma is the N side in this apparatus, and it is the stroma in which the ATP is released. Okay. Now coming to the dark reaction, which is also called as C3 cycle, Kelvin cycle, Kelvin-Menzel-Basham cycle, reductive pentose phosphate, that is RPP cycle. Sometimes we get questions in CSIR net with different such names, and you must be aware of them all, right? So through the light reactions in the chloroplast, we have produced ATP and NADPH. These two molecules will be utilized in the Kelvin cycle, thus releasing ADP and NADP plus, regenerating for light reaction again. But this will be utilized in order to fix carbon dioxide and convert into the glucose molecule. Okay, let's see the detail of this particular process, that is Kelvin cycle. This particular process doesn't require light, but it happens, doesn't need to happen at night only. It's just that it is independent of light, hence the name dark reaction. So Kelvin cycle occurs in the stroma, especially in where the ATP and NADPH are getting released. And these are the three important stages of Kelvin cycle. First is carbon fixation. That means the carbon dioxide will get attached with the molecule called as ribulose bisphosphate. So this molecule is a 5-carbon molecule, and carbon has one more. So totally you will have 6 carbon molecule. Now 6 carbon molecule will be broken down into 2 3-carbon molecules. Okay, the stoichiometry is little bit different because we have 3 molecules of each. Totally it will produce 6. I'm not going into the stoichiometry today. It will go beyond the scope. If you want some more details, you can attend Biotechnica live classes on CSIR net. Our new batch will be starting soon for the CSIR next CSIR exam. You can enroll for the same. So we have the molecule where we have 3 carbon. It is called as phosphoglycerate. 3-phosphoglycerate because one phosphate group is attached over here. Now we have the use of ATP. So 6 ADP molecules will be used, ATP molecules, one for each of the molecules, and another phosphate is attaching here. So from 3-phosphoglycerate it will form 1,3-bisphosphoglycerate, right? Now from 1,3-bisphosphoglycerate we have the NADPH which is used over here. They will give the electrons to 1,3-bisphosphoglycerate. PIs will be released and this is going to be reduced now. So from acidic component it will get reduced to form the aldehyde component. This aldehyde component will be further utilized to make glucose from glyceraldehyde 3-phosphate, but not all of the molecules. For example, if you have 6 molecules of glyceraldehyde 3-phosphate, 5 of the molecules will be regenerated for another round, whereas 1 molecule will be going for the synthesis of glucose. So till this phase, this is the reduction phase. The fixing of carbon dioxide was done by the enzyme Rubisco. It is a very important enzyme. This phase is called as carbon fixation, and from glyceraldehyde 3-phosphate till RUBP, it involves multiple of steps with multiple of enzymes, which is called as the regeneration phase. Again, today I will not go into detail of this, but this is how again RUBP is regenerated with the use of another 3 molecules of ATP. And this is the summary of all the reactions that we have seen here, starting from the light reactions, electron transport, ATP synthesis, and using the finished products of light reactions in the fixation of carbon dioxide and synthesizing glucose in the dark reaction. That's all for today. Thank you very much.